Microbiology and germ theory. In ancient times, people didn't know what caused disease. During wars, armies would catapult dead bodies and infected corpses over castle walls to infect enemies. The Romans, however, made a leap in public health by building sewer systems to remove waste. The word sanita, meaning health, reflects this. Still, people misunderstood illness. Many believed in bad blood, and sick patients were often bled in hopes of removing sickness, a practice that made things worse. Diseases often began in port cities. Rats carrying fleas would get on ships. The fleas would bite sailors, and infected sailors would carry diseases inland. The Black Plague was nearly 100% fatal back then. Today, it's about 25%. One symptom, swollen lymph nodes in the groin, armpit, or leg. Smallpox likely originated from cowpox or camelpox. When a disease is constantly present in a region, it is called endemic. 1. What does sanit mean? Health. 2. What is an endemic disease? One that's constantly present in a region. 3. How did the plague spread inland? Fleas infected sailors, and people fleeing carried the disease. Cell theory and microbes. Robert Hooke first used the word cell after viewing cork. Antony van Leeuwenhoek improved lenses and discovered live microbes. This started the field of microbiology. Cells come in two types. Eukaryotes have a nucleus, animals, plants, fungi. Prokaryotes, no nucleus, bacteria. Cells are measured in micrometers, millimeter. Viruses in nanometers, nanometer. Anything smaller than 0.1 millimeters can't be seen without a microscope. Quiz time. 1. Two main types of cells. Eukaryotic and prokaryotic. 2. Which has a nucleus? Eukaryotic. 3. How are viruses measured? In nanometers. Pasteur, Koch, and germ discovery. Louis Pasteur showed that microbes spoil food using a swan-necked flask. If the neck stayed intact, broth stayed clean. If broken, microbes got in. Robert Koch developed Koch's postulates. 1. Microbe found in all sick, not in healthy. 2. Isolate and grow microbe. 3. Inject into healthy host causes disease. 4. Re-isolate same microbe. Limitations. Some microbes can't be cultured and we can't test on humans, e.g. leprosy. Quiz time. What did Pasteur prove? Microbes come from the air. 2. What do Koch's postulates prove? Microbe causes a specific disease. 3. A major limitation? Can't test on humans or grow all microbes. Surgical hygiene and Lister's disinfectant. Surgeons in the 1800s didn't wash hands. Joseph Lister changed that by using phenol spray during surgery. Infection rates dropped. Phenol was toxic but led to modern disinfectants like Lysol. 1. What was the surgical problem in the 1800s? No hygiene. 2. What did Lister use? Phenol spray. Why isn't phenol used today? It's toxic. Cholera and epidemiology. John Snow in 1850s London mapped cholera cases. He found they clustered around a water pump. He removed the handle and the outbreak stopped. This began epidemiology, the study of how diseases spread. 1. What causes cholera? Bacteria. 2. What did John Snow do? Removed a contaminated pump. 3. What is epidemiology? The study of how diseases spread. Viruses. What makes them non-living? Viruses are smaller than bacteria. They are not made of cells, don't metabolize, can't reproduce on their own. They hijack host cells to reproduce. The first virus discovered, tobacco mosaic virus. Quiz time. 1. Why aren't viruses alive? No metabolism, no cells, can't self-replicate. 2. How do they reproduce? By hijacking a host. 3. How are viruses seen? Electron microscope. Vaccines. From cowpox to rabies. In the 1700s, milkmaids with cowpox didn't get smallpox. Edward Jenner created the first vaccine. Louis Pasteur made the rabies vaccine by weakening the virus in ducks. In the US, rabies still exists in bats. 1. How did cowpox protect from smallpox? Triggered immunity. 2. Who made the rabies vaccine? Louis Pasteur. 3. What U.S. animal still carries rabies? Bats? Antibiotics and resistance. Alexander Fleming discovered penicillin. It killed bacteria without harming human cells. This is called selective toxicity. Overuse led to antibiotic resistance. Bacteria with plasmids, extra DNA loops, can carry resistance genes and share them. 1. What is selective toxicity? Kills bacteria, not humans. 2. What causes resistance? Overuse of antibiotics. 3. What are plasmids? DNA loops that carry resistance genes. Normal flora and microbiota. We are born microbe-free, but soon we pick up microbes from air, skin, and our surroundings. These are normal flora or microbiota. They help with digestion, block harmful microbes, support immunity. 1. Are we born with flora? 2. What do they do? Help digestion and immunity. 3. What is microbiota? Microbes that live on in us. Classification of life. All life is classified: domain, kingdom, phylum, class, order, family, genus, species. Example: Escherichia coli, E. coli. Some strains are harmless, others are deadly. Quiz time. 1. 2 most specific taxonomic ranks: genus and species. 2. Format of scientific names: genus capitalized, species lowercase. 3. How can E. coli strains differ? Some harmless, some harmful, organic molecules and Kansas. Life needs 6 key elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Main macromolecules: proteins, amino acids, carbohydrates, sugars, lipids, fatty acids, nucleic acids, DNA, RNA. Water is also essential. Life is water-based. 1. What does COMP stand for? CHONPS. 2. What are the 4 macromolecules? Proteins, carbs, lipids, nucleic acids. 3. Why look for water in space? Life needs water. Macromolecules and protein folding. Proteins, carbohydrates, and lipids are built from monomers. Proteins must fold correctly to work. Heat or acid can cause denaturation, unfolding the protein. Example: cooking an egg. Quiz time. 1. What are macromolecules made of? Monomers. 2. What causes protein function? Proper folding. 3. What is denaturation? Unfolding of proteins. Gram staining and cell wall structure. Gram-positive, thick peptidoglycan, stains purple. Gram-negative, thin wall plus outer membrane, stains pink. Gram-negative bacteria are more dangerous due to endotoxin. 1. Gram-positive color, purple. 2. Gram-negative structure, thin wall's outer membrane. 3. What toxin in gram-negative? Endotoxin, mycobacteria, and acid-fast walls. Mycobacteria have mycolic acid in their wall. This makes them hard to treat. Examples: TB, Mycobacterium tuberculosis; leprosy, Mycobacterium lepra. Treatment lasts 6 months to 2 years. 1. Why hard to treat? Waxy wall blocks drugs. 2. TB and leprosy bacteria, Mycobacteria. Treatment time, 6 months to 2 years. Membranes and peptidoglycan. Bacterial membranes, phospholipids plus proteins. Walls, peptidoglycan, gives shape and prevents bursting. Penicillin destroys peptidoglycan. Lysozyme in tears and saliva also breaks peptidoglycan. 1. Membrane made of? Phospholipids and proteins. 2. What does peptidoglycan do? Keeps bacteria from bursting. 3. What breaks it down? Penicillin and lysozyme. Bacterial shapes and flagella. Shapes: coccus, round; bacillus, rod; vibrio, curved. Spirillum, spiral, equals motility; motile equals can move; non-motile equals no movement. Quiz time. 4 shapes: coccus, bacillus, vibrio, spirillum. 2. What helps movement? Flagella. 3. What does motile mean? Can move. Endospores and capsules. Endospores, survival form, resist heat, bleach, radiation. Capsules, sticky sugar layer, protects from immune system, helps stick to surfaces. Example: Streptococcus pneumonia uses capsule to avoid white blood cells. 1. What is an endospore? Dormant survival form. 2. What do capsules do? Protect and stick. 3. Why is S. pneumoniae dangerous? Capsule avoids immunity. Fimbriae, DNA, and plasmids. Fimbriae equals attached to surfaces, e.g. E. coli and UTIs. Nucleoid will hold circular DNA. Plasmids carry resistance genes. Can be shared between bacteria. 1. What do Fimbriae do? Help stick to. What's in the nucleoid? DNA. 3. What's in plasmids? Resistance genes. Metabolism and energy. Metabolism equals all reactions in a cell. Anabolism builds molecules. Catabolism breaks down molecules. ATP equals energy currency. Pathway. 1. Glycolysis. 2. Krebs cycle. 3. Electron transport chain. Without oxygen, anaerobic respiration or fermentation creates bad smells. 1. What is metabolism? All cell reactions. 2. Anabolism versus catabolism. Build versus break. 3. What carries energy? ATP. End of lecture. You did it. Let me know if